Dear learners, welcome you all in the course of Immunology. The course code is BZYET 141. Immunology. Basically, it is a elective courses. Uh, it is for the BSc General. Dear learner, today I want to discuss very important topic of immunology, that is known as the antigens. It is a very important. You will learn number of the things related to this topic. What is the expected learning outcome of today's session? So you will be able to define the antigens. What is the antigens? And after finishing of this session, you will be able to list the structure and properties of the antigen, and you will be able to differentiate between alloantigen and autoantigen. There are the two type of antigens are there, alloantigens and autoantigens. After finishing of this session, you will be able to differentiate between the T and B cell epitope. So it is a very important part of the antigens. After finishing of this session, you will be able to explain the small molecule can become immunogenic. Uh, After finishing of this session, you will be able to define adjuvant, heptane, and discuss their function and importance. So my session is divided into two parts, part first and part second. In the part first, we will discuss what is the antigens and classification of the antigens, properties of the antigens, and we will discuss different type of the antigens are there. While in part B, we will discuss in detail the super antigens, and various type of the adjuvant heptane we can utilize it, and another thing we will discuss in detail the B cell and T cell epitopes. So I want to discuss one by one. In my part first, what is the antigens? Basically, antigens is a part of molecules which can induce the production of the antibodies are the T cells. That they are able to produce the antibodies and are the T cells, and it again involve into the response of the immune or production of the antibodies, or it can engage to the destruction of the antigens like virus and bacteria. And antigen also called the immunogen are allergens that are present on the surface of the pathogen or cell. Basically, in case of the pathogens, are some cells are there, they have certain type of the molecules on their surface, they act as an antigen. If you take example of the bacteria, there are the several molecules which are present on the surface of the bacteria like lipopolysaccharide. They act as an antigen. So several proteins, several sugar molecules, they several lipids molecule, they act as a antigens. It can be part of their coat, capsule, cell wall, flagella, fimbriae, and toxins. So it is a part of the antigens are there. They are involved in the antigenicity of the particular molecules. It is made up with the complex molecules like proteins. So your antigen that is made up with the proteins, it is made up with the peptides, or it is made up with the polysaccharides, and sometimes nucleic acids and lipid along with the carrier proteins and polysaccharide can act as a antigen. So these are the antigens are there. It may be protein, or it may be polysaccharide, or it may be lipid molecule, or it may be combination, or it may be nucleic acid. They act as a antigen, and several other molecules which are present on the surface, they act as a antigens. So these are the antigens are there. Another thing I want to discuss the properties of the antigens. So there are the various properties of antigens which are available on the surface of the pathogens. The antigen from related species are less antigenic than the unrelated species. If you compare the animal and human, their behavior it would be unrelated, their behavior it would be different. The antigen from the different species or the less related species are the less antigenic than the unrelated species. So you can say it is a foreignness. Only the foreign molecule can evoke the immune response. Like when the pathogen infect to the human, then antibodies which recognize the antigens and begin rise into the blood. And phylogenetic distance between the species generate display between the antigens. So it is a foreign, it is a properties of the foreignness. Foreignness means related species are the less antigenic, and phylogenetic distance between the two species generate disparity between antigens. Only foreign molecule they are able to produce the antibody. So you can say it is a foreignness, it is a very important properties of the antigens. Another thing I want to discuss the molecular size of the antigens. If you see the molecular size, the minimal molecular weight 8 to 10 kilodalton to act as a antigens. So your antigen, their range, the minimal molecular weight 8 to 10 kilodaltons, they act as a antigens. The strong immune response fall in the range of 14 kilodalton to 600 kilodalton. So it is a range of your antigens. The size of the molecular weight of the antigens are there. Their molecular weight is 14 to 600 kilodalton, and less than 8 kilodalton molecule do not much weaker immune response do not or much weaker immune response will be generated. And serum albumins from the minimal mammals 69 kilodaltons is fairly good antigen. Hormone angiotensin 1031 daltons is a poor antigens. So you can see the size of the you can see the molecular weight of the antigens. It would be different, but their immune response it would be different. Less than 8 kilodaltons antigens do not or much weaker immune response will be generated. And 8 to 10 kilodalton they act as antigens, and the strong immune response will be generated. Their range it should be 14 kilodalton to 600 kilodalton. So it is very important characteristic of the antigens, their molecular weight. Another thing I want to discuss, it is another properties of the antigens. Uh, uh, what is the chemical nature of the chemical nature and composition of the antigen? The complex physical structure of the antigen are more immunogenic than the simple antigens. If you compare with the molecules, their complex physical nature of the antigens are the more immunogenic than simple one. If you compare with the primary, secondary, tertiary, quaternary structure of any molecules are there, the primary molecules it would be the less immunogenic. Compare with the quaternary molecules. The polymer of the proteins, polysaccharides, lipids, and nucleic acid can act as a antigen. And peptide antigen contain high number of the hydrophilic and charged amino acid like lysine, glutamine, glutamic acid, asparagine, aspartic acid. They are involved in the generation of the immune response. Another thing I want to discuss, the aromatic radicals can impart greater immunogenicity of the biomolecule. bacterial lipopolysaccharide. It is a very important part of the bacteria which are present on the surface of the gram negative bacteria, that is known as the lipopolysaccharide, are a good antigens. So it is a chemical nature and composition of the antigens. It may be protein, it may be lipid, it may be nucleic acids are there. They act as a antigens. Another very important natures are there, or you can say the chemical composition, like LPS, lipopolysaccharide, you can consider as a good antigens. Another thing I want to discuss, the physical form of your antigens. The soluble antigens are less immunogenic than the particulate antigen. If you compare, if your antigen is soluble, you can say it is a less immunogenic. If your antigen is not soluble, it is a particulate, so it may be more immunogenic. The molecule which are lost their 3D structures, it become more immunogenic. You can take example of the proteins. If it you can heat it, you can inject inside to the body, it may be more immunogenic than the native proteins. Another very important property, that is known as the antigen specificity. Antigen which are very specific. Antigen have unique site which impart them the uniqueness of the antigen specificity. These are called the antigenic determinant. The immune response is totally depend on the antigenic determinant or epitopes, how much epitopes which are present, and their quality of the epitopes which are very important. They are able to generate the immune response, and these are the site where that form to the bonds with the antibodies. The antigen specificity is a very important part of your antigens. You can say it is a epitopes, how much epitopes are there, what is the quality of the epitopes are there, it can decide the immunogenic response inside to the body. Another property is species specificity. It is another very important property of the antigen are there, and each species has tissues that where unique kind of antigens which are called the species specific antigens. It can produce unique antibodies when introduced into another species, and you can say it is a species specificity. Human blood protein can be differentiated from the animal protein by a specific antigen antibody reaction. You can just compare. It may be same protein, but the protein, it may be animal origin or plant origin. We can differentiate with the help of the specific antibody antigen antibody reactions. So we can distinguish which one is what is the specificity of the particular antibodies are there. We can check it, it is a human proteins or animal proteins are there. Another very important property, that is known as the organ specificity, and some antigen are unique to the particular organs, and these antigen can be similarly found on the organ of the different species. Thus antigen are said to be organ specific. The number of the antigens are there, they are the organ specific antigens are there, and which are present in the particular organs. It may be either animal or either, and the different type of the animals are there, like humans and goat, and their liver, they have the specific antigens are there. So you can say it is the organ specificity of the antigen. Autospecificity, it is another very important property of the antigens are there. The antigen present in the self tissues normally do not elicit the immune response, do not produce the immune response. In a disease condition, and they may evoke immune response and produce autoantibodies is known as the autoantigen. So your specificity, it may be autospecificity are there. So autoantigen, autoimmune response and autoantibodies will be produced. Another very important properties of the antigens are there, which are the genetic factors, and every species do not respond similarly to the antigens, and some species may produce an immune response against the antigen but other may not. It is due to the genetic makeup. Genetic makeup of the particular antigen, it may be different and they can maintain it. A species may lack or alter the gene that code for the receptor of the antigen onto the B and T cell. It is the region their antigen behavior, it would be different. So it may be it is another very important properties of the antigen, that is known as the genetic factors. Their genetic makeup, it would be different. And another very important properties of the antigens are there, that is known as the degradability. Antigen that are easily phagocytized and can produce better immune response. When the any antigens, it may be bacteria, it may be viruses, they infect inside to the body, it is easily phagocytized and can produce the better immune response. The phagocytosis is a very important phenomena for production of the immune response. The development of the immune response require that to the antigen phagocytized, processed and presented to the helper T cell by antigen presenting cell. So it is a phenomena which are very important. They are for the immune response. The inert molecules such as the dust, which cannot degrade inside to the body, produce allergic response rather than immune response. So it is a very important properties of the antigens. Your antigen, it should be degradable, it should be phagocytized, then immune response will be generated. Strong, you can say the strong immune response will be generated after infection of the infection of the pathogens. Another very important factors, another very important property of the there, that is known as the age. Ability to generate immune response is dependent on the age. So it totally depends on the age. The children under the age of 12 or elder beyond the age of 16 are less immunogenic than those age 13 to 16. You can see it. Their age, it is very important factors for production of the immune response are there. 12 or elder, they produce they are their antigens, it may be less immunogenic. Then those age 13 to 16, they produce the more immunogenic response will be there. Doses of the antigens, it is another very important properties of the antigens. The amount of antigen exposed to the body determine the immune response. So what is the quantity, what is the amount of antigen it need to expose to the body, then immune response will be generated. The exceptionally low dose or extremely high dose may not produce an optimal response. If you injected high dose and low dose, it is not produce the optimal response. Only appropriate dose can trigger an optimal response. So it need to decide it. It is a matter of the research. It need to check it what is the amount of the particular dose of the antigens are there. They are able to produce the very much immune response are there. So it is another very important properties of the antigen. What is the dose of the particular antigens are there? They are able to produce much immune response inside to the body. And another thing, the route of administration of the antigens. So what would be route of the antigens we can administer inside to the body? The antigen can be administered by the various route. It may be intravenous, it may be subcutaneous, or it may be intradermal are there. The subcutaneous and intradermal route give better response. If you inject your vaccine or antigens by the subcutaneous or intradermal route, it gives better response than the intravenous route. And the antigen administered intravenously is carried first to the spleen, and while the subcutaneous move first to the local lymph node. So it is a region you can say the subcutaneous and intradermal route give the better immune response than the intravenous route. So if you injected your antigen intravenously, it is carried first to the spleen, while the subcutaneous move first to the local lymph node. So it may be local lymph node, it may be generate a strong immune response. The subcutaneous and intradermal route give better immune response than the intravenous route. Another thing I want to discuss the type of antigens. Based on these epitopes, the antigens can be classified as the alloantigen or autoantigens. And another things it may be classified. First of all, it need to classify the exogenous antigen, endogenous antigens, then is alloantigens and autoantigens. So I want to discuss one by one exogenous antigen. You can see the structure of the microbes are there. It is a microbes. It is their DNA are there. It is a you can understand it is the antigens. It may be protein. It may be spike proteins are there. It is a microbes. It may be virus are there. The antigen can enter into the body by the ingestion, inhalation, or injection. The antigens, it may be virus, it may be bacteria, it may be fungi, or any other antigens are there. It can enter into the body by the ingestion, inhalation, or injections. And it is non self antigens, means outside of the body are there. So it is non self antigens and occur into the body fluid and extracellular space. It is present along with the MSC first molecules. So your antigen it will absorb through the cells and inside to the body, or it will process and present on the MSC second molecules. The example bacteria, virus, allergens like pollen and toxic food. And some antigen start out an exogenous antigen but later become the endogenous. Example the intracellular virus. The intracellular viruses are there. First it act as a exogenous antigen, and after that after some times it become change into the endogenous antigens. So it is a example of the exogenous antigen. It may be bacteria, it may be viruses, or it may be pollen or toxic food are there. Another is the endogenous antigens. Endogenous antigen are originate within the cell, like number of the cells inside to the body, like kidney cell, liver cells are there. The endogenous antigen are originate within the particular cells are there, and they are the self and non-self antigen present in the cell membranes, and which are presented along with the MSC class first molecules. And endogenous antigen are processed by the macrophages, and which are later accepted by the cytotoxic cell. Macrophages are APC, antigen presenting cell, which are later accepted by the cytotoxic T cell. By product of the regular cell metabolism or molecular component of the pathogen inside infected cell blood group antigens. So it is a example of the endogenous antigens. It can encountered by the cytotoxic T cell or it is another very important things here is the MSC class first molecule which are involved in case of the endogenous antigens. And third one is the alloantigens. The alloantigens are antigens which are found only the same member of the same species. Example, antigen A and antibody B present into the blood cells, RBC cells. They have, you can understand here is the RBC, they have the antigen and antibody. They have antigen A and antigen B present into the RBC. Here is the antigen A, antigen B. Here is without antigens are there. You can say it is the O blood group. Here is the A blood group. Here is the B blood group. Another is the AB blood groups are there. So here is best example of the alloantigen is the RBCs. They are determined genetically. So your genetically it is determined. They are also known as the isoantigens. Alloantigen produce an immune response non-self antigen from membrane to the same species. Blood group antigens are histocompatibility antigens are two major type of the alloantigens are there. So it is a best example of the alloantigens are there, that is human RBC. And another is the autoantigen. Sometimes in the autoimmune disorder, the antibody can be produced against the self antigen is known as the autoantigens. Under the normal condition, autoantigens should not be the target of the immune system, but due to genetic or environmental factors. Normal immunological tolerance has been lost of these patient. Nucleic acid and nucleoprotein are there. It is example. Example is the Hashimoto thyroiditis antibodies produced against the antigens and of the thyroid glands and lead to the gradual destruction of the thyroid glands are there. So it is a autoantigens. So in this case, the antigen can produce against the self antigens. You can say it is a autoantigen. So in this case, in case of the Hashimoto thyroiditis disease, so in this case the thyroid glands lead to gradual destruction of the thyroid glands. Dear learner, I want to summarize part first. You have learned the all molecule cannot provoke the immune response by producing antibodies. And you have also learned only self or alloantigen which are complex organic molecule with molecular weight is less than 8 kDa can be more than 8 kDa can evoke the immune response. And sometimes antigens, self-antigens, autoantigens can act as a foreign molecule due to the structural change and trigger to produce antibody against themselves. And you have learned to antigen can be classified as exogenous, endogenous, autoantigen, alloantigen based on their function and origin. Dear learner, we will take a short break. We will come within 2 minutes and don't go away.
Do you wish to be part of a television studio like this or any other media system? Or already part of any media system, want to enhance a new set of skills? Welcome to School of Journalism and New Media Studies. Here in the School of Journalism and New Media Studies, we are offering program for all set of people to learn and enhance their media skills. The vision of School of Journalism and New Media Studies is to generate a new set of professionals in the area of media and communication studies with understanding about role of media in the national development and global understanding. We offer innovative programs in the field of journalism and mass communication that sets the benchmark of high quality learning and teaching process at the national level. We offer these programs across the country with the help of our regional network as well as study center. Same manner, many of our programs are offered through online mode with a sophisticated learning management system. SOGNMS offers programs from PhD in journalism and mass communication till certificate level. We have masters in journalism and mass communication. The eligibility is a bachelor's degree, and we are introducing this program in 3 languages: Hindi, Tamil and English. Students from national and international students can join either through online mode as well as in the distance mode. We have 4 postgraduate diploma programs, specialized areas in the field of development communication, digital media, electronic media and journalism and mass communication program. And all these 4 programs are available in distance mode. 2 programs are available in online mode, development communication and digital media. And we have specialized certificate program in community radio. IGNOU employs various media and communication tools for better teaching and learning process. School of Journalism and New Media Studies utilizes this media system for delivering all our programs. Come and join with us for exploring a new career and enhance your media skills.
Dear learner, welcome you all in my next part, part second. In this part, we will discuss detail about to the super antigens. What is the super antigens? Basically, super antigens are class of antigen that are capable of evoking non-specific T cell response bypassing normal antigen processing. So, basically normal antigen processing will not take place in case of the super antigens. So, it can evoke non-specific T cell response. You can see this structure here is the T cells are there, here is the antigen presenting cells or dendritic cells are there. So, here is there are the chains are alpha beta chains are there. It is the MSC class first and MSC molecule are there. It is super antigen. It bind the super antigen is bind. It will bind to the beta part of your T cells and alpha part of your antigen presenting cells or dendritic cells are there. So, it is a situation where the super antigen will work or it will bypass the normal antigen processing or it evoke the non-specific T cell response. These antigens are usually, these are the super antigens are there. These antigens are usually protein produced by pathogen to combat against the immune system. Super antigen are more potent than normal antigens. Means you can say super antigen, they are able to produce the a lot of T cells are there, cytokines are there. Ultimately, shock will take place, and you can say it is a more potent. Super antigen are more potent than the normal antigen. Binding of the super antigen to the T cell receptor result the T cell proliferation and massive release of the cytokines. So after binding of the super antigens, the T cell proliferation and massive release of cytokines will take place. When the immune systems encountered a conventional T cell dependent antigen, only a small fraction of T cell population is able to recognize the antigen. You can see it if bind to the super antigens, only a small population of T cell they will recognize the antigen, and number of the antigens are there, they are not recognized by T cells, and but some antigen polyclonally relative polyclonally activate to large fraction of T cell are known as the super antigen. I want to give example. Staphylococcus enterotoxin uh, and Staphylococcus pyrogen exotoxins are example of the super antigen. It give the shock which are very dangerous to the humans. So these are the toxin, they are produced by the particular pathogens. Produced by particular pathogen, they produce the toxin. Their toxin they act as a super antigen. This super antigen they produce a lot of the T cells are there, and cytokines are there, or ultimately shock will take place, or um, uh, shock will take place with the help of the super antigens. Another very important things are there. I want to discuss the classification of the antigens. The antigen can be functionally divided into two groups. One is the complete antigen, another is the incomplete antigen. The complete antigens can elicit immune response on their own, called the immunogens, and complete antigen are generally high molecular weight and may be protein or polysaccharides are there. The complete antigens they are elicit, they can elicit the immune response on their own cell called the immunogen. They produce a lot of antibodies are there. There is no need of the heptanes and adjuvants are there. So you can say the complete antigens they are able to produce a lot of antibodies, and T helper cells are there which are able to provoke the immune response. Ultimately, they will remove the pathogen. They will destruct the pathogens are there. While in case of incomplete antigen, it cannot evoke an immune response till they complex with the proteins. Incomplete antigen are called the heptanes, and the protein which they are complex is called the carriers. So it is a two type. The if you classified of the antigens, it is a two type. One is the complete antigen, another is the incomplete antigens. Another thing I want to discuss the epitopes. What is the epitopes, which are very important? Epitopes are the site of the surface of the antigen where antibody binds. The antigen has antigenic determinant are the immunologically active site called the epitopes. Antibody form are specific to these sites, and it bind to the antigen specific membrane receptor, or the cells of the immune system are secreted antibodies. The epitope can be primary, secondary, tertiary, or even quaternary epitopes are there. So you can see this structure. It is a microbes. It may be viruses are there. They have the several very important part of the viruses are there. So it may be epitopes. The antibody will be generated against the particular epitopes are there. Which are found on the surface of the pathogens are there. So against the particular epitope the antibody will be generated. The antibody has antigenic determinants are immunogenically active site called the epitope. So these portion only particular portion are there. So it may be other portion are there, it will be generated another antibody. So you can say it is another epitope, not the circle like structures are there, it is epitope. But in spherical structures of the viruses are there, it may produce another epitope are there. Antibody form are specific of these site. So your antibody it would be produced which are specific to the particular epitopes are there. It bind to the antigen specific membrane receptor of the cells of the immune system or secreted antibodies. And epitope can be primary, secondary, tertiary, or even in quaternary structures are there. So here is antibody, they are very specific, they can recognize the particular epitopes. So you can see it here is the antibody, it is a microbes are there, they have the particular epitope, they will recognize with the help of the antibodies. B and T cell can identify the epitope present on the antigens. So your B cell and T cell they can identify, they can recognize the epitope present on the antigens. The particular antigen they produce, they have it may be have number of the epitopes are there. The properties of epitope are different for the both cases, and T cell and B cell epitope it may be different. The epitope thus have the inversely accessible site on the exposed to the surface of immunogen. The T cell do not recognize any random antigen. So T cell it is very specific. They are not able to recognize any random antigen. It will recognize very specific antigens are there of your T cell. The T cell interact with a part of the antigens where is present by the special molecules called the MSC on MSC which are present on the APC antigen presenting cell or nucleated cell. So your T cell which are very specific, they can recognize it, the particular antigens or particular epitopes are there of your antigens. The properties of the B cell epitopes. So what is the properties of the B cell epitopes? The epitope must have the shape that complementary to antigen binding site on an antibodies. So they basically the shape the epitope must have the particular antigens are there, they have the number of epitopes. The epitope must have shape that complementary to the antigen binding site on the antibodies. The size of the epitopes should not be larger than the receptors. The epitope should not be larger, the size of epitope should not larger than the receptors. Epitope must comprise the hydrophobic amino acids. Epitope mostly comprise the hydrophobic. Your epitope they contain the hydrophobic amino acids are there. The hydrophobic amino acid are hidden inside and thus cannot be an epitope of B cell receptor or antibodies. The amino acid sequences on the epitopes. Suppose the epitopes are there, they have the number of the amino acid sequence of the B cell can be sequential or not sequential. The amino acid sequences of the epitope on the epitopes of the B cell can be sequential or non-sequential. In sequential epitope, the amino acid are the continuous fashion along the polypeptides, while non-sequential has residual form and different segment of polypeptide brought nearly folded conformations are there. The antigenicity of the B cell epitope is determined by the presence of the group of amino acid and sequence of the polypeptide chain of the protein surface is displayed. So it is a very important of the B cell epitopes are there. They are able to recognize the antibodies. The properties of the T cell epitopes. The T cell epitope are processed and presented on the APC through the class first and class second MSC molecule. So basically T cell epitopes it need to process and present of your antigens on the APC. The APC they process and presented antigens through the class first and class second MSC molecule. The receptor on the T cells cannot bind to the free peptides or antigen independently. The antigenic peptides which is processed by the antigen presenting cells interact with the T cell receptor to form the trimolecular complex of MSC peptide T cell receptor. So here is very important things are there. The trimolecular complex of MSC peptide and T cell receptor will be there. Then it will provoke the immune response. And T cell epitope presented by the MSC first molecule typically linear peptide with length range from 8 to 11 amino acids are there. So it is a various properties of the T cell epitopes are there. And ligand cell interaction and signal transduction, it is another very important characteristics of the B cell receptors are there. The B cell receptor complex which contain the immunoglobulins bind to the B epitopes, B cell epitopes of the antigens are there. The binding alone cannot signal any message. This binding alone cannot signal any message. The adjoining proteins activate the immunoreceptor present on these proteins, which B cell receptor bind to B antigen, and this signaling is enhanced by the co-receptor CD4 on the B cell. The ligand of cell interaction and signal transduction is very important. They can communicate to the signal for further processing of the signals are there. So ultimately they will generate a lot of signals are there, or it will produce the antibodies and T cells are there, or ultimately they will kill the pathogens. The T cell receptor recognize the T epitope presented by the APC on MSC molecule and bind to it, and they are associated with the other protein that is known as the CD3, which get activated and stimulate the immune receptors. And this signaling is enhanced by the co-receptor CD8 on the T cells, and this response from the immunoreceptor can be modulated by the inhibitory signals around. Thus it helps to regulate the immune response as per surrounding conditions. So the both thing is very important. TCR, T cell receptor, MSC molecules which are displayed the antigens. So these are they are able to make the trimolecular structures are there, or ultimately they will recognize it, it give the signal to the cells, or it will produce a lot of T cells and cytokines are there, or ultimately immune response will be generated against the particular pathogens. B and T cell epitopes. So you can see it the B and T cell epitopes. Here is the T cells, here is the T cells are there, B and T cell epitopes. Here is the T cells. It is another the dendritic cells are there. Here is the protein antigens are there. Here you can see it is blue color, it is antigens. Here is the liquid antigens are there. The It is antibodies. Antibodies are there. Here is the cell membrane, or you can say the plasma membranes are there. They have the antibodies are there. So it is B cell and T cell epitopes are there. The T cell bind to the T cell epitope. T cell receptors and bind to the T cell epitope available into the antigen presenting cell and dendritic cell. So it is a dendritic cells. Here is the dendritic cells are there. Here is the T cells are there. Here is the protein antigens. Here is the liquid antigens are there. The B cell have Ig antigen immunoglobulins bound to the surface. You can see here is the cell membranes are there. They have the antibodies, and adjoining the protein of the B and T cell, which with the MHC of the cells help to the signal transductions. So after binding of this, they are able to produce the signals, or it will they are able to produce the cytokines, and they are able to produce the T cells are there, or ultimately they can kill the pathogens. Another thing I want to discuss, the heptane and adjuvant. The heptane are small molecules less than 1 kilodaltons, which contain low and low and cannot produce or the immune response. So less than 1 kilodalton, they are not able to produce the immune response. You can say it is the heptanes. Heptane are antigenic but not immunogenic. So heptane alone, they are not able to produce the antibodies, but it is a antigenic properties are there. When these molecules are linked with the larger molecule like proteins, carrier proteins, they evoke immune response. And the term heptane is derived from the Greek word heptane, meaning the fasten. And the carrier protein which are properly used are the albumins. The carrier protein, we can utilize it. It can combine with the heptanes. The heptane, they are able to produce the antibodies. So you can say it is a immunogen. So heptanes are the antigenic but not immunogen. They are not able to produce a lot of antibodies if you inject alone heptane, not with the carrier protein like albumins. You can see this structures, the antigens are there. The antigens, it is a two type. One is the low immunogenicity, another is the high immunogenicity. You can inject low immunogenicity in the organism, antibody will be produced. So their antibody, it will be very less, because your antigen, it is a low immunogenicity antigens are there. They are able to produce antibody, but their response will be very less, or you can say the less immunogenic of your antigens are there. Another is the high immunogenicity. You can inject inside to the any organisms, they are able to produce antibody, but here the immunogenicity, it would be very high. So it need to improve their immunogenicity of the particular antigens are there, or you can say the low immunogenicity antigen we can improve with the help of the heptane along with the adjuvant. Here is example of the heptanes. The various chemicals we can utilize as a heptanes. Dinitrophenols we can utilize as a heptanes, and aminobenzenes we can utilize as a heptanes. ortho aminobenzoic acid, meta aminobenzoic acid, para aminobenzoic acid, penicillin. So these are the heptane molecules are there. We can utilize it. We can combine with the low antigen, low immunogenicity antigens are there, or ultimately we can improve the production of the antibodies. Another is the adjuvant. An adjuvant is a pharmacological or immunological agent that improve the immune response to an antigen. It can be used with the antigens like vaccine or heptane to improve its ability to produce the immune response. The commonly used adjuvant are the inorganic compounds like aluminum hydroxide, alums we can utilize as a adjuvant, and mineral oils we can utilize as a adjuvants, and we can utilize the paraffin oil, and killed bacteria we can utilize as a adjuvant. So you can see in this figure, here is the cellular proteins are there, another is the carrier molecules are there, heptanes are there, it is carrier molecules are there, another is the adjuvant, and antibody can be developed by the heptane plus conjugate carriers proteins along with the adjuvant. So we can, in case of the vaccines, and we can utilize the various heptane adjuvant molecule. Ultimately, they are able to produce a lot of antibodies. Dear learner, I want to summarize my sessions of the part seconds. You have learned number of the things uh, only a small portion of the molecules which are called the epitope and bind to the B cell and T cells. You have also learned the mechanism of the binding of the T cells, how they bind it. Um, and you have also learned to the responses produced of the antigenic binding to the immune cells intensified the immune response leading to the antibody formations. You have also learned the sometimes molecules, small molecules called heptane along with the adjuvant added to the body fluid as a drug, analgesics or by injection can trigger antibody production. Dear learner, if you want to study in details, you can go through the IGNOU SLM BZYCT 141 unit number 6 antigens. It is a very important part of very important unit of your immunology courses. If any question related to this, you can mail me at as crm1 @ignou.ac.in. Thank you.
पहाड़ों पर जाना किसे पसंद नहीं। इस तरह के पहाड़ तो आपने देखे ही होंगे। चलिए आज आपको विशेष प्रकार के पहाड़ों की सैर कराते हैं। ये हैं कूड़े के पहाड़, जिनके बोझ तले दुनिया दबी जा रही है। इन कूड़े के पहाड़ों में हैं हमारे